Okay, hi everybody, happy Tuesday. Today is a pretty simple day in the fact that what we're going to be doing is taking everything that you've learned and you're going to be applying it to a situation. All the way back to even doing the activities, those learning opportunities before spring break. So you're going to see, you're going to have a chance to check to verify that you actually do understand citral dogma. That means protein synthesis. In other words, you understand that process. So we're making sure that you have all these connections because you're going to be doing an assessment on 512. So the understanding of genetic variation, central dogma, natural selection, all of that's going to play a role, okay? Our daily goal today, similar to the other days, only right now we're going to say that you're going to apply an understanding of protein synthesis. So go back to that central dogma, and you're going to see how you can use it to explain how mutations may or may not create genetic variation in a population. And if you'll notice, we keep emphasizing the idea of population. And the reason for that is an individual itself cannot change. You are already been made. So your genes are already been set and your phenotype is being expressed from whatever your genes are. And so it's important to realize that individuals do not cause natural selection. What's going to happen is the population, because of natural selection, is going to evolve and change. Your homework will be a worksheet. It's a molecular genetics. And you can see there on the Canvas page, I've already given you an attachment to it, okay? All right, so I can use scientific reasoning to develop an explanation for the case of evolution and natural selection that identifies, explains, and predicts changes. That's the main thing that we're doing. You'll notice the same vocabulary word. The only things that we've added to it are substitute mutation, insertion mutation, deletion mutation, a silent, a mutation, a missensus mutation, and a nonsensus mutation. Okay. How do both mutations and protein synthesis create variation within a population? Obviously, the activity is going to be all wrapped around that. Okay, so again, this activity is going to be dealing with the rock pocket mice. Okay, so we're just reminding you a couple things you already knew. When you did your graphing of location A and your graphing of location B, most of you had trends that look like this. You can take a moment, you can see where you've been able to make sense on how changes in the environment may cause changes to which traits are most commonly expressed. Okay, again, it's not going to cause the mouse to say, I think I'd rather have a dark coat. They can't do that. They have what they have, okay? But for those of them that had the more appropriate color, they had a greater potential survival. That's just the bottom line, okay? So a key concept so far, and these are the big ideas, these next two slides, is the fact that natural selection is a mechanism. It's the driving force for evolution, the theory of evolution. Now, when you're saying driving force, what do you mean by that? Well, let's think back to another theory. If you think of the theory of plate tectonics, okay? What was the driving force that was moving the plates? Well, it was the convection cycles in the mantle, right? And so that's the driving force, that difference that we had in the heat, right? In the transfer of heat. So those convection cycles are what were driving the plates apart and close together. Well, here we're talking about what's the driving force of evolution. Well, that driving force or that mechanism is natural selection. And as we just discovered in a couple of the activities you've done so far, natural selection is a non-random process. And it's the result of, if you look at these next four things, if you notice what I highlighted in red, in order for a change in a population to be a result of natural selection, 4 factors have to be in place. I'm hoping to see those 4 factors when I read your CEJs. I'm still behind them. I'm still doing the number 9s. Okay. There must be variation. In other words, different traits within a population. Okay. Those traits must be able to be inherited. In other words, they got to be able to be passed on from one generation to the next. Okay. Those traits must cause differential survival or reproduction within that population. In other words, the environment causes one trait to have an advantage over another. Okay, and then of course finally that 4th factor or that 4th condition is that the change must persist over several generations. otherwise known as adaptation. Because when a population of organisms, either physical or behavioral features, change to function better in the face of an environmental pressure. That's the definition of adaptation. So an individual organism himself can't do it. It's going to require the population of that species, okay? Second part of the keys are mutations that give rise to variation in a population occur at random. Natural selection leads to adaptations as the frequency of traits and the genes that code for those traits change within a population over time. And many different sources of scientific evidence support claims that evolution by natural selection has occurred and is still occurring, okay? Unfortunately, the image on the right-hand side kind of got the top part here. This basically says population of mice over time. And so you can see this could possibly what the variation looked like. And over time, it could change to this, okay? It's just giving you an idea that it's the populations that change, not the individual. So important to catch that. So the types of mutations, again, a total review from what you saw before. There's a substitute mutation, that's the replacement of one nucleotide with another. Mutations that affect a single nucleotide are called point mutations. So here you can see what your DNA, oops, Daisy. Here you can see what your DNA would be, right? Notice how we have T's in there, okay? You don't have T's in the RNA, right? You're gonna have U's, okay? And so this are your DNA bases, okay? And so here you can see that, let's see, original DNA. This is the original DNA. And then there must be an mRNA that would have to have come forth, okay? So that would have been a G, a U, and an A, and a G, and a U, and an A, and a G, and a U, and an A, and a G, and a U, and an A, okay? This is cat. And then this amino acid we're gonna find out is based on what the mRNA, okay? The mRNA are codons, remember? The DNA are the code. mRNA are codons. If this doesn't show you the codons, but like I said, if this said GUA, okay, that would be the mRNA codons, and this would be the amino acid that that codon would represent, okay? Now they're saying to you, well, what would happen if in this DNA a single nucleotide got replaced, okay? So it should be an A for cap, but remember it got changed to a C. Well, then what's gonna happen is ultimately, then you're gonna have GGA, that would be the RNA, and then that would be a different amino acid, okay? Insertion mutation, that's when you have an extra, you either add one or more nucleotides, remember these are all bases, okay, to the DNA gene sequence. And the assertion of a nucleotide can result in a frame shift. In other words, it can go one in and then it's just an extra one which does nothing. Or it could be added in and then it causes these three over here where these two are now gonna go with that. And so this is now going to go with the next, the whole entire thing is going to shift, okay? A deletion mutation, that's the loss of one or more nucleotides from the DNA gene sequence. And the deletion of nucleotides can result also from frame shifts, okay? Your textbook, one of the readings you were supposed to have done a couple of times back, gave you a lot of information with regard to mutations. So don't forget your textbook's a great resource. Gene mutations, okay, so now we're gonna say, all right, so we know that the DNA, that's where your genes are, that's the genetic information, and a mutation can happen, and what does that imply, okay? Well, a gene mutation, as it says over here, is any change in the DNA sequence, okay? So let's say the DNA sequence is supposed to look like this, but then for some reason it looks like this, okay? Then it's no longer gonna have the exact same information as this one does to produce ultimately that correct protein, okay? And so what could happen is it's either gonna create an abnormal protein or no protein at all, okay? A gene mutation can change the structure of the resulting protein, okay, that's what I was trying to say, changes the structure completely. A change in a protein structure can change, negate, or in some instances it might even not have any effect, okay? There are several types of mutations and they affect the amino acid sequence of proteins in different ways. Okay? So if we were to tie this back, tie mutations back to protein synthesis, okay? If we were to tie this all back, We began this unit by learning about DNA mutations in central dogma, protein synthesis. And today, we're going to be tying these concepts back to our rock pocket mice example to answer the following question. How do mutations in protein synthesis work together to create variation within a population? And so we've already talked about how you can have different traits, okay? So we can see how the variation can be. This is called a widow's peak here. Okay, this person is able to not roll their tongue, but this person can roll their tongue, okay? Which thumb is on top? That's really interesting. I'm not too sure what that one is. Your length of your second toe, okay? So they're asking how long that toe is. Is it longer than your big toe or shorter? I'll look at mine real quick there. Mine is shorter. Your little finger is straight or crooked, and looks to me like it's pretty straight. Earlobes, we already know mine are attached, okay? The front teeth close together, yeah, mine are close together, okay. All right, so potential effects of genetic mutations on protein synthesis. When a mutation occurs, there are three main effects that the mutation can potentially have on the proteins that are produced, okay? One is called a silent mutation. That means no change, okay? So that mutation does not cause a change in the amino acid sequence. You're gonna find when you look at the codon chart, which is all of the codons, all the various variation of the three codons coming together, the RNAs, all the different ways they can come together, and you're gonna find that they all are coding for a specific amino acid. And yet some of them have a lot more possibilities, a lot more different ways to make that exact same amino acid. And so who knows what nature was doing when it made that opportunity. So sometimes, even if you do have a mutation, it might not even cause any issues, okay? Missensis mutation is a change in the amino acids. So this mutation causes an amino acid in the sequence to be changed to another amino acid, thus causing the change in both the primary and the three-dimensional structure of protein, okay? So definitely something different is going to occur. So if we look at these right now, we can see where the silent is saying that there's no change here, okay? So this was the DNA, this is the mRNA, that's the protein. Missensis, here we can say that this has been changed, okay? And when this got changed, okay, this is what you're going to get with respect to this. Now, it's kind of hard to see this since each one of those DNAs are different types, but hopefully you're getting the picture. All right, so nonsense mutation is a change in the protein length, okay? This mutation causes the protein to be truncated, meaning cut, shortened, due to incorporation of a stop signal. So something comes in that tells it to stop, okay? This results in a translation being stopped before the amino acid sequence of the protein is even completed. Therefore, you're not going to have all the necessary information to make that protein, okay? It's like taking a gigantic train with all the cars, and it's supposed to have 100 cars, but at some point at 25 cars, it cuts off, and so all that other information is missing from it. So the MC1R, we know that there's two pigments primarily determining the coat color of the rock pocket mice, okay? Eumelanin, which is a dark color, and phenylalanine, which is a light color. The MC1R gene encodes for a protein called melanocortin, which is melanocortin 1 receptor. That's what the MC1R stands for. I'd rather say MC1R, which is found embedded in the cell membrane, specialized in pigment production cells. So your melanocytes. The melanocytes of wild type, when you see the word wild type, it's kind of funny. You think that's the bizarre one. Wild type is just what's the non-mutated. That's what would be normally expected. Those are going to produce the phenylalanine, there we go, more than eumelanin. That would be a result in a sandy colored mouse, okay? The mutated version, on the other hand, will trigger the melanocytes to increase the production of the eumelanin and result in a dark colored coat phenotype. Whew, don't worry. It doesn't matter to me if you can't pronounce these, I'm not gonna know. Okay, So what are we doing today? All right, so the overall assignment, you're gonna see this next 6 slides pretty much, are all having to do with the molecular genetics of the color mutations of the rock pocket mice. You're gonna get more specific now. You're getting down all the way down to the nitrogen bases. Okay, so now you're gonna get down to those bases to find out what's going on. First of all, make a copy of the document. I'm not too sure that this one's gonna come up or not, but the one that's on Canvas that I've already given to you, it should be accessible. I forgot to unlink that. You're going to follow the directions on the next few slides on how to fill in the tables for questions 2 and 3. So we're going to go back and check to make sure that you actually do understand the process of protein synthesis. And then after that, you're going to fill in the table that's continuing questions 4 through 10. And then when you've completed it all, you're going to upload it to Canvas. Okay. So if we take a look at this, using the DNA nucleotide sequence for the wild type and the mutant genes in the table on pages 2 and 3, it's asking you to determine the complementary mRNA sequence in the yellow highlighted boxes, or just one box actually. Note, you're only transcribing short portions of DNA sequence for the proteins. In actuality, it contains over 954 base pairs. We're not asking you to do that, okay? So using the PowerPoint and the mRNA base pairs that have already been filled out for you to help guide you, okay? So you can see here, here's your DNA, right? TTG, your mRNA would be AAC. Oh, okay, so if this is GCG, the mRNA would be, what would it be, okay? Keeping in mind that DNA bonds with DNA, those are complementary base pairs, okay? But when the DNA bonds with RNA, RNA has uracil instead of thymine, okay? And so you can see here that when you have DNA, there is no thymine in RNA. So what's gonna happen is A will bond with, oops, Daisy. A is gonna bond with U instead, okay? And T will bond with A, okay? So you're supposed to figure out, first of all, what that mRNA would be, okay? All right. Next, using the mRNA sequence completed in step 2, all right, so you've got that figured out, and the genetic code chart shown below, determine the resulting amino acids. So now you're going to try to figure out what the amino acid should be. That's that whole entire sequence. So this whole sequence is what it would be. Well, this is a portion of the sequence. And you'll notice that these are all the abbreviations for the amino acids. The chart actually gives you the full name so that you can see if you would like to know what the full names are. So you're going to use, you're translating only a portion of the protein. Keep in mind, you're not doing the whole thing, okay? The full protein actually has 317 amino acids long. You're not doing that many, okay? You're going to use the PowerPoint, and the amino acids have already been filled in to guide you, okay? So you can see this was T, this was A, this was G, this was C, this was T, this was A. And so then using this, it helps you figure out what it is on the codon chart. Now, once you've transcribed the DNA to the mRNA base pairs, you then can use what's called the codon chart to figure out the amino acid that each mRNA codon will tell the ribosome to make, okay? Remember translation? Okay, so the next slide explains how to read that. And so if we go to the next slide, okay, so here is a codon chart, okay? And unfortunately, my picture is going to be in the way. I can get rid of it, okay. I don't know if it went away or not. If you look at this, you're going to see that this is base 1. So anything in the first column over here, notice how this is G, these are all the first. This is A, these are all the first. This is C, this is all the first. This is U, this is all the first, okay? Second base are these here. So U must be in the second one all the way down, okay? C must be in the second one all the way down. A must be in the second one all the way down. And G must be in the second one all the way down, okay? And then third base is broken up into these sections here, okay? So you can see U, U, and U, right? U, okay? U, U, and C. UUC, right? U, and A, right? See how that's done? Now, if we were to go back and say, okay, ACA, it looks like it's something with THRE, ACA. So we have A, okay, we have C, and we have A. There we go. That's how you got it, okay? And so this is the abbreviation for the amino acid that the codon codes for, okay? And like I said, this one gives you all of the names first. I apologize that for some reason this one here, this actually should belong, oh no, it all fits there. It's all perfect. My mistake. Okay. These are some extra resources to help you get prepared and to help you study a little bit. And so, again, keep in mind, I am just going back and looking over number 9, haven't even touched the CEJs. Had a lot on my plate this past weekend, so I didn't get to grade. Sorry about that. But I will be getting to the number 9s tonight and tomorrow and get those all posted for you, okay? So take the time to make sure you understand how to do all this. Tomorrow at 3 is going to be office hours if you have any questions. If not, again, this hopefully isn't hard for you. It's just kind of revisiting things and pulling things together and making sure that you have the basic ideas that are really super key, all those major concepts, okay? All right, you guys, have a great Tuesday, and that's it.